I've been a member of the Gairdner community for 12 years. My name's Philip Marsden, and I help run the review panel that starts the process of deciding who our Gairdner Award winners are for the year. It's been a proud affiliation, and I must point out that working with folks like Dr. Dirks' leadership and Dr. Rossand as the new leader has been one of the most exciting aspects of my career. I wanted to give a few minutes on the review process because we are transparent about the review process. The nominees and the reviews themselves are confidential, but we want to be transparent and not opaque about how we select the finest. There are two panels, a medical review panel made up of rising stars, 25 to 30 of them in any given year throughout Canada, and we get on average, in the last few years, hundreds of nominations. Last year, for instance, there were 160 nominations, many that were new, and the first process of the review panel is for these rising stars in Canadian science to say, who should we put right to the top? From that, a short and a long list is developed. Sometimes the applications need a bit more finesse. This is a great area, this is the right subject. We have this name, that's great, but do we have all of the names? And that's a complex process in today's science because, as our speakers in the earlier part of the morning have told us, science is a team effort nowadays. There's a second panel, the Medical Advisory Board. Many members are here this afternoon. Dr. Sheckman, for instance, a former Nobel laureate. And the purpose of that is to willow down this list of these 160 initial nominations and to come up with an idea as to who ranks right at the top at this moment in time. Sometimes we see nominations for a few years until the science is crystallized into something that the Gairdner family, when the awards first started in 1959, were key. Does it make a difference in human medicine? Sometimes that's a hard judgment. Early technologies take a bit of time to develop into those that are practical. Some things go right to the top very quickly. One can think of how quickly the RNA interference pathways were recognized. And the Gairdner Foundation in 2005 recognized Fire and Mello, and the Nobel Prize followed shortly after. That review process we're very proud of. We think it serves us well. It's often worth looking at what we're doing and how we do it, but we're all very proud of the process. This afternoon, we're going to have the opportunity to hear three of our laureates in this year. Following on in the theme of the total 5 awardees in the field of CRISPR-Cas9, our next speaker is going to complete this story. Dr. Feng Zhang, I would have loved to have been on your promotion committee when you got promoted to associate professor this year. You know, for those who sat on promotions committees, sometimes it's nice to have an easy package. For the students in the audience, I just want to highlight that Dr. Feng is a bit younger than me. I've got that disease of aging, but the ability to do high-quality science and be recognized early in your career is out there. He emigrated from China to the United States, Des Moines, in 1993. And what's really interesting is that he got his first interest in science starting as a high school student in someone doing genome editing in his home city. He quickly went on to excel in his studies, went on to do a Ph.D. work with Disroth in the optogenetics field, an exciting area that many scientists follow with avid interest. But in 2011, he opened up his own lab, where he's a member of the Broad and McGovern Institutes. And today he's going to talk to us about the next stage of the gene editing paradigm and story we've heard about. He's got about 24 trainees in his laboratory, and it's amazing to see his young career flourish as it's had. I would love to be on your next promotion committee.
Good afternoon. It's truly a tremendous honor and a tremendous pleasure to be here. I want to first thank the Gairdner Foundation for this tremendous honor, and I had the opportunity to visit Alberta, Edmonton earlier this week, and I'm really I feel quite fortunate to be able to see Canada and interact with the scientists throughout the country, meet not only high school students but also researchers and learning about what they work on. It's truly been a tremendous pleasure, and I hope that we'll continue to have many more interactions in the years to come. So I'll tell you a bit about sort of application of genome editing for various areas. And just by way of getting started, to date, there are over 6,000 genetic diseases that have been identified. And we know the genetic mutation that underlie many of these different diseases. So now you might think, if we know what the mutations are in the genome that cause these devastating illnesses, why don't we just go into these cells and then fix these genetic mutations? Why don't we just replace what is causing the problem with the wild type sequences? Well, it turns out that the genome is microscopic, and going into the genome, which is already quite complicated, it's 3 billion bases long, making precise changes in the genome is very challenging. And indeed, this is a challenge that has been worked on by many, many laboratories over the past several decades. One of the earlier approaches relied on the use of DNA binding domains, and it's a really nice idea where if we can build proteins that can be customized to recognize a specific DNA sequence within the genome, then we can use that protein as an anchor to bring nuclease domains to specific sites and then facilitate editing. Maria Jason and also Jim Haber discovered over 15, 20 years ago that if there's a double-stranded break within the genome, then those double-stranded breaks will be sensed by the cell as DNA damage, and it can trigger endogenous repair machineries belonging to either the non-homologous injoining pathway or the homology-directed repair pathway to stimulate and increase the rate of gene repair. And so using these DNA binding domain anchors, we can recruit nuclease domains to specific sites and make double-stranded breaks and then be able to stimulate an HEJ or HDR. And so the key thing here is really developing an easy-to-use ability to introduce double-strand breaks. And so here are two of the earlier technologies. So one of them has been worked on by many labs for more than a decade. It's called the zinc finger protein technology, where zinc finger proteins are naturally occurring protein motifs that recognize and interact with DNA. But through directed evolution or targeted mutagenesis, it's possible to alter the binding specificity of individual zinc finger modules. And you can link the modules together to form what we call a zinc finger array to be able to recognize a long stretch of DNA sequence. So each module of a zinc finger recognizes 3 bases, and if you want to recognize 9 bases, you will have 3 modules. 18 bases would mean you have 6 different modules. But the challenge here is how do we go and engineer the specificity of these modules? Turns out that's a very challenging task. So even if you have someone who is very deeply experienced in engineering zinc finger proteins, it can still take several weeks, and even then you're not guaranteed to have something that's easily and efficient for the target sequence that you're trying to modify. A few years ago, a second DNA binding domain technology surfaced, and that's the tail technology, which is in some ways analogous to zinc finger because it also uses individual DNA binding domains that recognize single DNA bases that you can link together to form a tail array. And these tail proteins can recognize long stretches of DNA sequences, 12 bases, up to 30-some bases at a time. But still, tail proteins are highly repetitive. Each module is more or less identical to every other module, so you're working with a long polymer. And working with that long homopolymer takes quite a bit of complicated molecular biology manipulation to generate new proteins. And so because of these challenges, one of the needs was to see, can we develop a new genome editing system that's not only easier, faster, but also cheaper so that we can really broadly apply it to study many, many different genes. So earlier this morning, we heard really a fantastic introduction by Phil and Rudolph on the basic CRISPR systems, and also by Jennifer and Emmanuel on the mechanism of the CRISPR system. So my foray into the CRISPR area began actually with a paper published by Sylvain Moyneux, who is a Canadian scientist at Laval University. And back in 2010, Sylvain published this paper where he showed that the CRISPR-Cas9 system uses RNA guide to be able to base pair with the target DNA and then facilitate targeted DNA double-strand break. When I read that paper, I got immediately very excited because as I was thinking about working on nucleases, especially I was working on tail nucleases at the time, when I heard about the ability to use RNA to guide the specificity, I thought this could be a much simpler system if we can get this to edit in the human genome. One thing to note about the double-strand break that's generated by Cas9 that Sylvain showed is that unlike the type 1 and also type 3 CRISPR systems, where type 1 not only cleaves DNA but also does more damage around the target site, and type 3 system is a transcription-dependent DNA endonuclease, type 2 system makes a clean cut, and then you can use that cut to stimulate an HEJ or HDR process. So at the time, there were many different applications that we heard about this morning, and a lot of it were focused on industrial applications. But I thought maybe we can use it, harness Cas9, so that we can edit the genome of eukaryotic cells, so that we can have an easier, cheaper, and also faster genome editing technology. So we worked on exploring different Cas9 proteins, and so we showed that you can take two different families of Cas9 proteins, one from S. thermophilus, the protein that Sylvain Moyneux had in his paper, and also another protein from S. pyogenes that Rudolph and Emmanuel talked about in their studies. And by combining these Cas9 proteins with the tracer RNA and also with a CRISPR array, and along with the host RNase molecule, we can use it to be able to introduce double-stranded breaks. And so we engineered the CRISPR array with a small guide sequence that matches the target site on the human genome in the EMX1 gene, and then it's flanked by a short motif called the PAN motif. And then using this system, we can use Cas9 and tracer RNA and also CRISPR array to facilitate genome editing within human cells. And so when you have all four components, or even without the bacterial RNase 3, you can achieve genome editing within the human cell. Another nice thing about the CRISPR system is that it's naturally multiplexed. So each CRISPR array carries multiple guide RNAs to target many different viruses. And one application of that in human cells is that you can use this CRISPR system's natural multiplexability to target multiple genes at once. And so we tested whether or not this would work. So we designed Cas9 and tracer RNA to work with a CRISPR array that carried two different guide sequences that target two different genes within the human genome, the EMX1 gene and also the parvalbumin gene. And then we showed that using this approach, we can simultaneously edit both genes within human cells. And so here, with the array containing EMX1 and parvalbumin, we can achieve editing at both of the loci in the genome, and then it's equally efficient as if we introduce the individual guides to edit one or the other locus. So just to recap, we can use Cas9 to facilitate genome editing, and we can use it in a couple of different ways. By taking these Cas9 family members from Thermophilus or from S. pyogenes, we can either use CRISPR arrays along with trace RNA to facilitate editing, or we can use a single guide RNA that we heard about this morning to facilitate genome editing. Using either methodology, we can stimulate an HEJ or HDR by introducing double-stranded breaks within the specific target site. So one of the really exciting applications of CRISPR-Cas9 is maybe we can one day develop this as a therapeutic to go and treat diseases. And we thought, can we try to see how well we can use this in an animal model of disease? Well, one of the challenges that we first had to resolve is how do we develop a Cas9 that we can deliver efficiently in vivo and we can edit with high efficiency to achieve a therapeutic benefit. S. pyogenes and also S. thermophilus Cas9 are two Cas9s, but they are large and sometimes challenge to package into a single viral vector so that we can deliver both the Cas9 gene and the guide RNA. And so we thought, well, there are many different Cas9s out there in nature. Maybe we can try to harness other Cas9s that are even more compact, that we can fit into convenient viral vectors and test its in vivo editing efficacy. So here is the phylogenetic tree showing the distribution of many different Cas9 family members. And you can see it's really widely spread through different bacterial species. If you plot the length of these Cas9s based on their amino acid length, you can see that they distribute into two different clusters. There's a short cluster that's about 1,100 amino acids long, and then there's a longer cluster that's around 1,350 amino acids long. And we thought, well, maybe we can find some of the shorter ones that really falls at the shortest tail end of this distribution. So we began by choosing 6 different Cas9s that represent this cluster is very, very small Cas9. And we eventually found one that can work quite well, and that's the one from Staphylococcus aureus. This protein is about slightly more than 3 kilobases long, and that means we can package both the Staph aureus Cas9 as well as its promoter and the guide RNA within a single adeno-associated virus. We chose AAV because it's one of the most advanced gene therapy vectors. So to see how well this would work, we then designed a guide RNA that targets the PCSK9 gene, because we thought if we can use a viral vector that delivers into the liver and then knock down or knock out PCSK9 expression, then we can reduce cholesterol, as PCSK9 is a very prominent drug target for reducing cholesterol and reducing cardiovascular disease risk. So to see how well this works, we injected these into mice systemically. So you can prepare the virus and inject through the tail vein. So we designed 2 different guide RNAs to target PCSK9, and we built 2 different viruses to see how well does it work. On the left side is the editing activity. And so this is the x-axis, so that's the time course. And so within 7 days after injection, we already saw quite good level of editing. So you get about approaching 50% of PCSK9 indels within the liver tissue section. When we sampled the liver tissue for subsequent time points, we saw that the editing activity is stable and it persists for the rest of the experiment. Now that we have Cas9, PCSK9 editing, how well does this translate into a therapeutic change in the animals? So we took serum from these PCSK9 knockout animals, and we measured the amount of PCSK9 protein residing in the serum. And the time course really agrees with the editing activity. So this is before the injection, and this is 7 days after the injection. And across 4 different dosages, so A-fold dose range, we saw that for each one of these injections, we no longer were able to detect PCSK9 in the serum of these animals. So a reduction in PCSK9 should translate into a reduction in cholesterol. And so then we next measured the amount of cholesterol in these animals. And what we found is that within 7 days after injection, there's also a 50% reduction in the cholesterol amount in these animals. And so these early data point to that the possibility that maybe we can develop this as a therapeutic tool to directly modify genes within somatic cell tissue or into cells that we have taken ex vivo, and then modify them to achieve a therapeutic outcome. Now, one of the challenges of advancing Cas9 to a therapeutic application has to do with how well can Cas9 modify only the target and not introduce any off-target mutagenesis. So over the past few years, we and also several other groups have very comprehensively analyzed the specificity of Cas9. And what we found is that Cas9 indeed can introduce off-target activity. And so that's not ideal, because when we go into a cell, we really just want to correct the on-target mutation, and we don't want to introduce other mutagenesis within the cell. So we took an approach to try to improve the Cas9 specificity. And there were work by us and also by many other labs to improve the specificity, but most of those approaches are fairly complicated. They required modifying the Cas9 enzyme to recruit other nucleases or modifying the RNA or changing the inducibility of the enzyme. So we thought maybe we can develop a simpler solution. And so 3 of my teammates, Ian and Ling Yi, and also Bernd, they looked at the way that Cas9 will bind to DNA. And they thought maybe we can tweak the way that Cas9 stabilizes non-specific DNA binding to reduce off-target mutagenesis. Here is a schematic of how Cas9 would bind to DNA. So DNA is shown here in red, is naturally duplexed. And in order for Cas9 to bind, that DNA has to be unwound. And then the RNA can base pair with the target DNA. So RNA is shown here in green. When the RNA hybridizes with the DNA, the displaced DNA strand is negatively charged and is not stable. And so in order for Cas9 to bind there for long enough to dwell there and cleave the DNA, this displaced DNA strand needs to be somehow stabilized. And so we hypothesized that maybe there's a positive surface along where this displaced DNA strand would fall, and we can try to weaken that non-specific stabilization to make Cas9 more specific. So here is the crystal structure colored in the same way as the schematic, but you can also color it a different way by looking at the surface charge distribution. And this is how that looks. Blue is positively charged and red is negatively charged. And so you can see this groove right here where the displaced DNA strand is supposed to fall is very blue. And so we thought maybe let's go into this region, inactivate some of these arginines and lysines and change them to neutral residues like alanine so that we can weaken the non-specific binding and then make Cas9 more specific. So we tested about 30 different mutants with either individual arginine lysine substitutions or with combinations of mutations. And we found that we can indeed make new versions of Cas9 mutants that are more specific. And so here is an experiment where we began with a wild-type Cas9 and a guide RNA that targets the VEGF-A gene. We knew from previous experiments that this guide RNA has two prominent off-targets. And you can see here is the on-target editing, and these are the two off-target editing sites. Now, with the enhanced Cas9, we're able to reduce it down to levels we're no longer able to detect using the specificity measurement assays. So both of these are what we call enhanced Cas9s with 3 different mutations, and they preserve the on-target activity while more or less eliminating the off-target activity. And so this shows that we can take a rational structure-guided engineering to make these enzymes more specific. We also did an experiment to see, are we really requiring more stringent base pairing between the RNA and the DNA to achieve editing? Here, this is with the wild-type Cas9, and the first bar shows a perfectly matching guide RNA. And then below that, each of the guide RNA shows pairs of mutations along the length of the guide. And what we found is that in agreement with many other studies, the 5' region is less specific, so they can be tolerated if there are mismatches. The 3' region is quite specific, so it doesn't tolerate these kinds of mismatches. This is what the data looks like with the enhanced Cas9. Now, because we are weakening the non-specific stabilization, that means the RNA needs to provide more stringent binding to the complementary DNA. And so here, even the 5' region requires base pairing complementarity in order to have editing. And so using both of these Cas9s, we can take advantage of full length of the guide RNA to achieve editing. So we can use structure rational guided engineering to make Cas9 better, but we are no smarter than nature. And nature, after all, has had billions of more years than us to evolve and invent new technologies. And so we thought maybe we can explore nature's diversity more. There may be other enzymes that nature has evolved that we can harness that would make genome editing more powerful. So one of the studies we did is to explore previously uncharacterized CRISPR systems. And if you look at why this might be interesting, we can look at the way that CRISPRs are classified. Broadly speaking, there are 2 major classes of CRISPR systems. There's the class 1 system, which utilizes multiple proteins that come together to facilitate the RNA-guided recognition and also destruction. Class 2 systems are more simpler. They use a single protein like Cas9 to facilitate recognition and also target destruction. Now, there are multiple class 1 systems, but at the start of our study, the only understood system was Cas9. And so we thought maybe we can explore other class 2 systems to see if they are also useful for genome editing. One of the uncharacterized class 2 systems fall in this organism called Francisella. It happens to have 2 CRISPR systems, one encoding Cas9, and then another one that encodes a gene called Cpfl. If you look at the domain organization Cpfl, you can see it's quite different from the domain organization of Cas9. And the domain organization for Cpfl is very similar to a lot of transposable elements. And so it wasn't clear whether or not a transposable element just happened to have landed right next to a CRISPR repeat system, a repeat array, or Cpfl is yet another CRISPR RNA-guided endonuclease. So we transplanted Cpfl locus from Francisella into E. coli, and we found that indeed is an RNA-guided endonuclease system. In fact, it uses a different RNA targeting substrate, whereas Cas9 is a dual RNA-guided system requiring both a CRISPR RNA and also the tracer RNA that Emmanuel told us about. Cpf1 uses just a single RNA guide, and it has a direct repeat that's on the 5' end flanking the target site. So we thought to see whether or not we can use this for genome editing, and we chose 16 different Cpf1 family members. We knew from our Cas9 work that not all Cas9s can just be plucked out of bacteria and put into a human cell. And out of the 16 Cpf1s, only 8 of them were able to get reconstituted in vitro, so they can cleave using a human cell lysate expressing these proteins. And out of these 8 that work in vitro, 2 of them worked in vivo to be able to achieve genome editing. And we were happy to have found any, so we decided to focus on these 2 Cpf1s from Pseudomonas and also Lactobacillus. Next, we thought to see how specific is Cpf1. And so we designed a guide RNA that can be targeted by both Cpf1 and also by Cas9. And we found using an unbiased genome-wide specificity assay called BLESS that whereas Cas9 exhibited 2 off-target sites, the Cpf1 system was more specific. So we didn't see any off-target cleavage. And if you choose a guide RNA that is unique in the genome, Cas9 can also be quite specific, so you don't pick up any off-target double-strand break activity. And Cpf1 also can be quite specific. And Keith Zhang and also Jin-Soo Kim's groups have also carried out analysis, and we have found very similar specificity enhancements. Another unique thing about Cpf1 has to do with the way that RNA is processed. And so Emmanuel told us that Cas9 uses tracer RNA to process CRISPR array into the individual dual RNA guides. And then Emmanuel also found that Cpf1 processes the CRISPR array into the individual RNA guides. And this is advantageous because this means that we can even better achieve multiplex editing. We can just use a single promoter to drive a long cRNA array that carries multiple guide RNA sequences. So we tested to see whether or not this would work, and we built 2 different AAVs, one that expresses Cpf1 using a neuronal specific promoter, and a second AAV that uses a U6 promoter to drive a triple array guide RNA that can get processed by Cpf1 and also a GFP marker just so that we know which neurons got transduced. After we inject it into the brain, we can get very good transduction in the dentate gyrus of mice. These are red fluorescence that are stained from Cpf1 expressing cells. And then the green fluorescence shows cells expressing the expression of the second guide RNA vector. And so there's very good co-localization. And if you dissect out that piece of brain tissue and measure the level of editing, you can see that there's very good editing at all 3 genes. And so for MECP2, neuroligin 3, and also DRD1, we can get editing in these postmitotic neurons just through a single tandem array expression. We have continued to mine these CRISPR systems because Cpf1 is unlikely to be the last CRISPR system to be found. And so we set out a really fantastic collaboration with Eugene Koonin and Konstantin Svirinov, where we used a computational pipeline to identify novel CRISPR systems. We began with a protein called Cas1, which is the most conserved CRISPR enzyme involved in the acquisition of immunity. And then by doing BLAST, we can pull out all of the bacterial sequences, all of the contigs that carry the Cas1 gene. And we can annotate the open reading frame so that we know what are the nearby genes. And because CRISPR systems are encoding the operons, we had good expectation that the nearby genes are likely to also be CRISPR-associated enzymes. We can then annotate each protein as either an unknown or a known gene, and we just focus on unknown genes because they would point us to novel CRISPR proteins. And so using this pipeline, we were able to identify a number of different new candidates. And so for each one of these candidates, we thought to characterize them more and see whether or not they have interesting potential as new novel technology. So here is Cas9 and Cpf1. Using this pipeline, we were able to pull out 3 new CRISPR candidates. That's Class 2 Candidate 1, Class 2 Candidate 2, and Class 2 Candidate 3. And the most interesting one to us was the Class 2 Candidate 2, because unlike all the other enzymes, it contains nuclease domains that are found normally in RNases or in toxins. And so we thought maybe this is an RNA-guided RNA targeting system that we can use to target a different set of experimental applications. Based on that hypothesis, we then set up a screen. We chose the RNA-only virus, MS2, which is a phage that only exists in RNA form. And it's a small genome, only 3,800 bases, so we can design guides that tile along the entire length of the genome, shifted by one base at a time. And by cloning these guides designed and synthesized in vitro, you can put them into vectors and transform E. coli. And now each E. coli carries a single guide RNA that complements with the MS2 phage. So by challenging these bacteria with MS2, we can see, do any bacteria survive? And if any survive, then that means some of the C2C2 system might be active, might have defended the E. coli against the MS2 infection. And moreover, from sequencing the remaining guide sequences, we can figure out how does C2C2 target its target RNA. So based on that experiment, we found that for the Leptotrichia shahi C2C2, the targeting requirement just that the guide sequence target site has to be flanked by a single base. It can either be a C or A or a U. So this is great. Now we have a RNA-guided RNA targeting system, and we're trying to now explore its various applications. So one of the things we thought to see is, can this enzyme have activity in eukaryotic cells? And so we modified this enzyme so we can express it in human cells. And then we compared it with using shRNA to knock down a luciferase target gene. And so for two different target sites, this is the first guide and this is the second guide, the cRNA conditions compared favorably with shRNA conditions. And we can use it to be able to knock down gene expression. In addition to using C2C2 for knockdown experiments, we can also use it to just bind to RNA and try to visualize things. And so very much like Cas9, we can inactivate the hairpin domains to turn it into a generic RNA-guided RNA binding protein. And so here we fuse this inactive C2C2 with mCherry so that we can bind to mRNA in cells and then visualize the localization of this mRNA. To test this, we used a cRNA that targets the beta-actin gene. So in collaboration with Alice Ting, we transfected all these into cells and applied a stress condition, sodium arsenide, on these cells. And when the stress was applied, we saw that mCherry fluorescence localized to stress granules perinuclear. And if you don't add the stress, then the mCherry expression is entirely cytoplasmic. And this shows that we can use this as a way of monitoring and tracking RNA localization in living cells. So now we have a lot of different tools. We can perturb DNA, we can perturb RNA, but we also need to develop readout methodologies to figure out what are the perturbations doing. Without reading it out, we won't really advance our understanding of the biological mechanism. And so toward that end, we've been developing single-cell, single-nuclei sequencing methodologies so that we can pair that with editing to figure out what is happening in these cells. And so Naomi and also Inqing worked on developing a technique called NucSeq that we have applied to neuronal tissue, because neurons are intrinsically very elaborate and have complicated morphology. And so by taking out only the nuclei from neurons, we can more easily extract out cells and then carry out individual cell sequencing. We found that the transcriptome from a nucleus and transcriptome from a whole cell are fairly representative of the transcription state for the cells. And so from doing this type of experiment, we can carry out functional classification of cells. So by doing completely unbiased clustering analysis on the individual nuclear transcriptome, we can group hippocampal tissue from the brain into different cell types from different brain regions, dentate gyrus or CA2 or CA1, or interneurons within the hippocampus. And we can also do subclustering too. So you can take the GABAergic cluster and then divide it into parvalbumin cells, somatostatin cells, and many other different kinds of cell types. So we applied this to see, can we use this to study a biological process? And we combined it with EDU-based labeling to look at proliferation of newborn neurons within the adult hippocampus. And so using this methodology, we can label and then collect cells at different time points after the initial labeling, and then look at how does transcriptome change at different time points post differentiation of these cells. And so we use FACS sorting to extract out these labeled nuclei. by doing the same analysis, we can cluster them along a very nice temporal trajectory. So this is all of the cells, and different colors represent different groups of cells extracted at different time points after the initial labeling. And you can see that they fall along this continuum. So the blue cells are 1 to 2 days, and then these really dark red and kind of orangish cells are from the later labeling time points. So based on this time trajectory, we can then identify what are the marker genes that are expressed at specific time points. So for example, by looking at expression for SOX9, we find that it expresses in the early post-differentiation cells, whereas NeuroD1 expresses in the later post-differentiation cells. And so by getting a time course of how the transcriptome change, we can then use some of these marker genes to potentially drive the maturation of cells into specific fates. And so we can especially combine that with in vitro cellular models that we generated using genome editing to be able to study functions of specific mutations. So this is pretty much it. Just to recap, we are really excited to continue to explore the natural diversity and these interesting microbial systems to be able to develop novel technologies. And we are also committed to developing the applications and also making sure that these tools are easily accessible and distributing it to other laboratories. And then as the tools become more mature, we're also really excited to start to apply it to the neurological questions that motivated us to develop the tools in the first place, and to really see if we can advance our understanding of some of these diseases. So as we continue, we're continuing to take census, and this is the latest census from CRISPR systems. So in addition to the class 2 and also the Cpf1, C2C1, and also the C2C2, the C2C3 and C2C4 systems, and also the type 6, we most recently computationally identified yet another RNA-guided RNA targeting system that we call Cas13b, and we are now just starting to functionally study what these systems are doing in bacterial cells. But this is really the most exciting and inspiring thing. This plot shows the number of genomes, bacterial genomes that have been completed over the past 15 years. And you can see in the past 5 years, the number of genomes that have been completed has really grown exponentially. And this really represents unprecedented opportunity to go and harness and understand, explore these diversity and harness them to be able to further advance our ability to study biology and also to be able to treat diseases in the future. So finally, this is the last slide, but also the most important slide. This is my team. The development of these technologies has been a team effort. And it's really thanks to many of the students and postdocs who I have worked with, shown here, and also our long list of really fantastic collaborators that we have been fortunate to work with to develop many of these technologies and applications. And last but not least, many of the funding agencies, both governmental and also private foundations that have supported our work over the past several years. Again, thank you again. I want to thank the Gairdner Foundation again for this opportunity, and it's really a great pleasure to be here.